Dina, oh Dina, where are you? DNA structure and replication by Miss Hoskins. All right, so for DNA structure, you're going to learn how to analyze genetic information, how it's expressed in cells, and then we want you to be able to explain how the structure of DNA and even RNA are going to lead to the expression of information in the cell, and that is through a process of replication, transcription, and translation. Nucleic acids. Here's the ultimate macromolecule again. This macromolecule holds our genetic information, and that is called DNA. DNA stands for deoxyribonucleic acid, and it is a wonderful and huge molecule. It contains genes. Genes are little sections and pieces of this DNA that serve as the instructions, or almost like a blueprint, if you will, of making proteins. These proteins basically drive all cellular activity. Now, there's two different types of nucleic acids. DNA, or deoxyribonucleic acid, and then RNA, ribonucleic acid. They have different structures, and we'll talk about where they're found and why their structure is a little different. So the monomer, or the smallest component making up nucleic acids, is a nucleotide. And if you remember, it has three different parts: a sugar, a phosphate, and a base. So the sugar, which is represented here in blue, can either be deoxyribose or ribose sugar. Depending upon what sugar it is, we're going to have RNA or DNA. The phosphate group right here is represented in red. And then these nitrogenous bases can come in 5 different forms. We can have adenine, thymine, guanine, cytosine, and one that is particularly special is the uracil, which is only in RNA, right? So uracil is only in RNA, and thymine is the one that is unique to only DNA. DNA is a double helical structure. It was discovered by Watson and Crick in the early 50s. 1953 is when they're accredited for coming up and designing the structure of DNA. Even though it was a woman, Rosalind, who actually, sacrificed her own well-being through using radiation and X-ray technology and came up to discover that it was helical in structure. It was actually Watson and Crick who took modeling pieces, put it all together, and sold it as a double helical structure, and so they received the Nobel Prize, and of course they received credit for the DNA structure. Okay, that's just a little history lesson there, but it's a double helix, so it's a twisted ladder twisted with another ladder. The sugar and the phosphate, they form the backbone of this ladder. So every sugar is connected to the next nucleotide's phosphate. So the phosphate, sugar, that kind of forms what we call the backbone of the DNA molecule. And the nitrogen bases, those are in the middle, and they form these weak hydrogen bonds. Remember, hydrogen bonds have an electrostatic attraction, so they're kind of they just hang out near each other really, really closely. They don't really exchange electrons and stuff like covalent bonds that share electrons, or ionic bonds that transfer electrons. They don't really do that. They just hang out really, really close to each other. And of course, all the other bonds that are in this whole system are considered covalent bonds because they are non-metal and non-metal type of bonds. Now, if we look at these nitrogenous bases, their complementary pair in the hydrogen bond has a match. The complementary pair for adenine is thymine, and for guanine it's cytosine. So all of the A's, they're going to pair up with T's, and all of the C's, they're going to pair up with G's and vice versa. So wherever you see a guanine on a DNA molecule, opposite hydrogen bonded to it is going to be a cytosine. And same with adenine and thymine. If you have a thymine, you're going to see its paired up hydrogen bonded with an adenine, and all the adenines will be paired up with the thymines. And this bonding of the nitrogen bases together creates this latter type of structure. We call these complementary base pairs. And because they bond specifically together, they are complementary base pair rules that say A bonds with T, C bonds with G. So at this point, that's where we sing the DNA song, and it is to the theme of Row, Row, Row Your Boat. We love DNA made of nucleotides, sugar, phosphate, and a base bonded down one side. Adenine and thymine make a lovely pair. Cytosine without guanine would feel very bare. Applause! Yes, I'll be here all week. These nitrogen bases have two types. Adenine and guanine, we call those the small words, but they're a big base. Those are called purines. And then the pyrimidines, which is a big word, but it has a smaller base, are cytosine and thymine. Here's what I mean. The purines, adenine and guanine, it's a smaller word. Purine's a smaller word, but it's a bigger base. Pyrimidines, that's a really big word, but it's talking about the smaller bases, cytosine and thymine. Chargaff's rules imply that adenine and thymine pair up and cytosine and guanine pair up. They're held together through these weak hydrogen bonds, and they hold together in pairs. Now, adenine double bonds to thymine, and then cytosine has a triple bond to guanine. This pattern of the same amount of adenine as thymine found in a strand of DNA applies to both strands of DNA, along with cytosine and guanine. Chargaff is the one that determines that this rule had to have this 1-to-1 ratio. Now, how he came up with that ratio, that stoichiometric ratio rule of purine and pyrimidine, is beyond the scope of the course. If you're interested, take AP Biology, you'll learn all about it. But for now, we just want you to know that adenine and thymine, they form these double bonds, and cytosine and guanine, they form these triple bonds. And these are the rules and how they're paired. They're hydrogen bonded in the center of the helix. DNA is also anti-parallel. That means the strands are running in opposite direction of each other, or they're like turned upside down. One strand is running what we call 5' to 3' and in that direction, and the other strand is running in the 3 to 5' direction. Phosphate is always at the end of the 5', and then deoxyribose, the ribose sugar, is always at the end of the 3'. So 3' is related to the sugar, and 5' is related to a phosphate. And so what I mean by that is that the complementary strand is running sugar to phosphate down here. It's flipped and running anti-parallel, as it's called. This 5' and this 3' have to do with the sugars that are on deoxyribose. They're actually, numbered 1, 2, 3, 4, 5, and so that's what they're talking about, is whose number is kind of sticking out over here, 3, and then 5s. But in any event, you need to know that they're running anti-parallel, the strands of DNA. Now at this point, we want you to pause and practice. So pause and practice labeling the parts of DNA. How did you do? Were you able to label the DNA molecule as seen here? Now it's RNA's turn. RNA is a single strand of nucleotides, and so it has the bases, the nitrogen bases, they're exposed. And so RNA has, it binds with DNA's base, but then what happens is that the A's are going to bind with U's and the C's are going to bind with G's. So RNA has no thymine. In the place of thymine, we have added uracil. Everywhere on the DNA strand where there would have been an adenine, and so that would normally have bound with, this is the DNA strand, that would have bound with thymine. When we are making DNA and we're going to RNA, we're going to have a uracil in place of the thymines that would have been there complementary. So there are some differences between DNA and RNA that we should note. DNA has adenine, thymine, cytosine, and guanine, while RNA has adenine, uracil in the place of thymine, cytosine, and guanine. The sugar that's there is a deoxyribose sugar in DNA, and it's a ribose sugar in RNA. The shape of DNA is a double helix, and in RNA, it's a single strand. The basics of heredity lie in DNA. Chromosomes are tightly coiled strands of DNA. Different organisms, they have different number of chromosomes. Like humans, like we have 23 pairs, so that's 46 total. 23 we got from mom and 23 we got from dad. Dogs actually have 37 pairs, so that's like 74 total chromosomes, 37 from mom and 37 from dad. Some strawberries now, they have like 400 strands. So really, depending upon the organism, will depend upon the number of chromosomes that are there. A gene is a little section, it's a little piece of that DNA, and it has instructions to make a protein, a special protein unique to that organism. On one chromosome, you can contain thousands of genes that are all linked together that make all these different proteins to build this complex body. So genes, they're basically like sections and pieces of DNA where along chromosomes, which are long strands of DNA. And chromosomes are like all bunched up. So I love this diagram because it attempts to explain some vocabulary here. So this is a chromosome, and if you unravel it, you'll see that it's a chromosome that's all bunched up. And as you unravel it, it's DNA. It's a strand of DNA, and it is coiled on a double helix. So there's actually technically two strands there that are coiled. If I unravel it further, then it will uncoil itself into a single strand here. And along the lines, you will start to see all of the base pairs. So the reason we can get so many genes inside of one tiny little cell is because it coils up tight inside of the nucleus to form these chromosomes.